Hi everyone, my name is Amit. I finished my PhD a few months ago, and I'll be presenting one of the projects I work on my PhD. And just letting you know, this is the first time I'm talking about technical aspects of my PhD. So I did my MSc in physics and computer science, and then I moved on to biology. So I'm used to talking about my projects with biologists, and they're not computer scientists. So I needed to simplify things. So I hope I won't get stuck describing the technical things, and just bear with me. So I'm going to talk about signal processing on genomic data, and just to give you a little bit of a feeling of the sizes of data and what I'm working on, we need to first talk about what the data is. So I'm working on what you might know as a genome, but actually there are two things that we usually talk about when we refer to genomes. We have a genome and we have an exome. So a genome is everything that defines who you are. It's the entire sequence of your chromosomes of any species or any creature's chromosomes. So it's the entire sequence. It's about 30 billion base pairs, okay, 30 billion letters. And if you think about it from a data point of view, how do you save this data? And each character is 1 byte, so it's 8 bits. You get about 24 gigabits of data. That's a lot of data for one genome. An exome is just 1% of the genome, and it's all the letters, all the sequences, all the words that would eventually translate into functional units, which is called proteins. So it's actually what's making you live. And as I said, it's 1% of the genome, so it's about 30 million base pairs, which is about 240 megabits. But we don't save just the sequence. We have to save some sort of annotation. And annotation is something that tells me something about what this gene, this part of the exome does. So it's the name of the gene and the name of the protein that would originate from that gene. It's the location of the gene. It's the strand that the gene is coded on. It's, I don't know, all sorts of annotation data that is saved, which creates a file of about 1.5 gigabits, which is large for one exome. So each and every one of you is coded by either 24 gigabits of genome data, 1.5 gigabits of exome data. And I'm going to show you an analysis that I did on 20 exomes at once. So it's a lot of data. Just as a small note, when you do your home kits for genome sequencing, it's usually not your entire gene. Okay, so don't let the big companies confuse it. Okay, so just going back to biology 101, DNA is the sequence that is encoded in our chromosomes. DNA is transcribed into what is called RNA. Both DNA and RNA are sequences of letters that are called nucleotides. We have 4 types of nucleotides, and some of the RNA would be translated into protein. The RNA that is translated into protein is called mRNA, messenger RNA. I'm going to focus on this part, okay, the part that is translated to a protein. And how does translation happen? So we have this mRNA molecule. It needs to be translated. It's translated by a machine called the ribosome, which you all need to know. So the ribosome would recognize an mRNA via some signals. It will bind to this mRNA, and it will start traveling along this mRNA until it encounters a start codon. It's a start signal. It's 3 nucleotides. Okay? This is the signal that tells it this is the location you need to start translating into a protein. So this is a location where another subunit of the ribosome would bind, and you see this little thingy here. This is called a tRNA. tRNA is what mediates triplets of nucleotides into one amino acid. So amino acid are the letter that codes proteins. So triplets of nucleotides would be translated into amino acid. You see these 2 slots of the ribosomes. So once the ribosome recognizes this initiator tRNA, it will follow to find the mRNA, the tRNA, sorry, that would match the next triplet. So it's the next codon. It recognizes it, it attaches it to its next slot, and a peptide bond is created between these two amino acids. And then what the ribosome would actually do is move this tRNA into its first slot, so now it is free to find another tRNA for the next slot that it can insert into. And this goes on until the stop signal is recognized, which is again another triplet, which is called the stop codon. This is where translation would end. The peptide would be cleaved from the ribosome, and now it is free to be functional in the cell. The ribosome will disassemble and would move on to translate another mRNA. This is the classical dogma of protein translation, and it has two very important features. The first is a non-redundant matter of translation. What does that mean? I told you that translation is done in triplets. Each triplet of nucleotide would be translated into one amino acid, and this is done in a non-overlapping manner. So the ribosome would translate one triplet, move on to the next triplet. One thing I failed to mention is when the ribosome recognizes its start signal, this location, this triplet, like this actual signal, would define what is called the translational frame. So what is the translational frame? It's just the location, the position, where you need to start looking at triplets of codon, and this is the way the protein is translated. So this is one property. Another property of coding is redundancy. So we have 4 letters for nucleotides, for RNA. Triplets are coding for amino acids, so this creates 64 possibilities, 64 combinations, but we only have 20 amino acids, which means that some triplets would code for the same amino acid, and we're going to go back to that in a minute. So these are the two main dogmas of classical translation, and I'm going to talk about non-classical translation. And I'm going to talk about a specific example, which is called ribosomal frameshift. So I told you that translation is done in a non-overlapping manner, but what happens if in a single location, in a single point during the translation, the ribosome would not move 3 nucleotides, would not move 3 nucleotides downstream, or would not enter a tRNA that matches the next 3 nucleotides, but it would move 1 nucleotide, or 2 nucleotides, or even move back. This would change the point of reference that was set for us from this the start signal that I talked about, and it would create a different protein that would have been if translation would have moved on in a classical manner. And this, if it happens, and it's not due to error, if it happens in a programmed way, this needs to be very regulated. Because if this happens in a non-regulated manner, it would cause a lot of mass in the cell. It could completely affect the protein's function because it would change its sequence. And actually, this mechanism happens a lot in viruses, which have a very small, compact genome, but they still need to code a large repertoire of proteins. And in mammals, there are a few examples, like a handful or 2 handfuls of examples of non-canonical translation, which were all discovered empirically, so not systematically. And in my PhD, I devised a method to systematically identify non-canonical translation originating from ribosomal frameshift. And it uses the fact that we have redundancy in the coding. So if you pay close attention to this redundancy, you would see that triplets coding for the same amino acid would usually differ by their third position.
You have 3 stop signals.
Yes.
Don't see a start signal.
The start signal is this one. It encodes for an amino acid called methionine. So the stop signal doesn't enter an amino acid, but the start signal does. And methionine, by the way, can happen during the sequence. So it marks the beginning of translation, but it's also a normal amino acid. So you might have methionines just floating around in the sequence. So again, we see that triplets coding for the same amino acid usually differ by the third position, and this allows for some plasticity in creating a protein coding gene. So you might have protein coding genes that are slightly different, but they still code for the exact same amino acid sequence. And we can utilize this property to actually create a periodic signal. So we would take sequences from different organisms, humans, chimp, mouse, whatever. In our case, it would be 20 species. And we would align protein coding genes that code for the same protein using bioinformatic algorithms that exist to get what is called the multiple sequence alignment. It's like a table of letters. And for each position in this multiple sequence alignment, we could devise a score for variability. So we would get, because the third positions in codons are less conserved, we're supposed to get a pattern of conserved, conserved, unconserved position along this multiple sequence alignment. And once we have a periodic signal, all the mathematicians here know that we can just analyze it using Fourier transform or some other harmonic analysis, and this is exactly what we do. So we're not using exactly Fourier transform because it's a biological signal. It's very noisy. It has a lot of regions without any periodicity at all due to high conservation. But we can use properties of Fourier transform to devise something more discrete that would only give us indication of the frequency that we're interested in, which is 1/3, because every 3 nucleotides, we're supposed to get the same pattern. So what we do is just take this multiple sequence alignment, create this variability signal, and we would devise 3 different magnitudes from this signal. We would take the variability score for each first subcodon position, sum it up. We would get one measure, all the second, and all the third. We would sum each one on its own to create 3 vectors on a 2-dimensional space. So these vectors, this vector is just the summation of all variability scores in the third position. This is a measure of all the variability scores in the first position, and this is the second position. And if you pay close attention before, you might have seen that very highly conserved positions have a negative variability score. What this translates into in this space is that the vector would change its direction in 180 degrees. So for a normal codon, on a normal protein translating gene, since it's supposed to be conserved, conserved, unconserved, the first and second position should change their direction, while the third should remain in its original direction. It should be very positive. And what we would do with these 3 vectors is just radially sum them. So we radially sum them to get one resulting vector, which has a magnitude and it has a phase. And we can use this resulting vector to infer the translational frame. This vector would actually point into the most variable position within a codon. Since we know that the third position should be most variable, we can infer the translational frame. And analysis is done in a sliding windows manner. So what you see here is the variability vector of a gene called hemoglobin. It's a very normal gene. It doesn't have non-canonical translation in it. If you look at this signal, which shows the variability per position, I don't know, maybe some of you can see some periodic pattern, but it's not very obvious. And I want to point out that I told you that the ribosome would bind to this mRNA, it would start moving until it encounters the start signal, and it would translate until it encounters a stop signal, which means that we have flanks on this mRNA. We have the flank before the start signal and after the stop signal, which shouldn't be translated into a protein. So they don't need to hold this periodic pattern. And in this variability plot, we see the entire mRNA. So we have these flanks. The flank before the start signal is called the 5' UTR, untranslated region, and the flank after the stop codon is called the 3' UTR. And you can, in your eyes, see and guess where they are. So analysis would result in a sliding window manner. We would analyze each window using the approach I showed you with the 3 vectors. So this plot shows actually the 3 vectors pointing in each direction. And each sliding window would move 3 nucleotides downstream to its predecessor to maintain the frame of reference. So this would go on along the gene. And you can see this region where the third position is most conserved, is most variable. And I told you that this is a normal translating gene, so you might assume that this is actually the sequence between the start and the stop, the coding sequence. what we do from these triplets of vectors, we would derive the magnitude I told you and the phase. Okay, so you see this these graphs, and from this we would infer translational frame, and we would get this plot. So we have a straight line in the canonical frame. It's very long. It would probably indicate on the coding sequence. Then we have these flanking regions, which are the magnitude measure helps us understand how many periodicities did we see, or how periodic is this sequence, the sequence really is. So it's kind of a measure of confidence for the inference of it, right? And indeed, the coding sequence is capturing the entire condition frame. This is classical translation, canonical translation. What happens in the non-canonical case? So this is a non-canonical case. This is a gene called antizyme. It's the only known case in mammals to have a ribosomal +1 frameshift. It's a big deal. I'm not sure you're getting how much of a big deal it is. It means that somewhere along translation, the ribosome would move 1 nucleotide downstream instead of 3, which is major. It needs to be very, very regulated.
Is it the same ribosome that?
The same ribosome. It's the same machinery. So this means that you have some sort of hints within the signal that might tell the ribosome, okay, now you don't need to nucleotides. I'm not going to talk about it today, but if you want, you can read my thesis. Okay, this is the variability score. Again, if you're very keen, you might see a periodic signal, but I bet you can't detect changes in the frame, in the phase. This plot, you can see clearly that the third position, which is the one pointing to the canonical frame, is switched by the first position. And again, in the phase graph, you can see a steady state being replaced by another steady state. And this is actually the inference of frame. So in antizyme, translation starts in the canonical frame, and in some location it moves to the +1 frame. The pink line shows the actual frameshift location, and we have an error here. The error is 6 codons, so it's 18 nucleotides. And the reason that we have an error is because we're working with sliding windows. So the fact that we have only 18 nucleotides error with a 135 nucleotide window, it's quite good. So this is one example, and we have one more example in mammals, which is PEG10, which has a -1 frameshift. So we see similar behavior. Error here is 8 codons, so it's 24 nucleotides. And we have 2 more examples from viruses. The first is from HIV virus. It's called the drug hole fusion protein. And it's very similar to PEG10. What happens here is that we have 2 proteins, 2 separate proteins that are translated together as one gene, and later they are cleaved. And the proteins are translated in different frames. So while the ribosome is translating one gene, it needs to frame shift to get the other gene. And we detect this frame shift, but there's something very special about this fusion protein is that there's a small overlap between the 2 genes. So there's a small region in this translation that should be able to be translated in 2 different frames. And if you look at the signal of frame shift here from the 0 to the from the 0 to the -1 frame shift, and you look at the signal here, it's different. And this difference is actually due to the fact that there's an overlap in here. Another very interesting observation that we see is from HPV, the human papillomavirus. The E2 locus, the E2 gene, has another reading frame, another gene encoded within it in a different frame. So this is not a case of ribosomal frame. The ribosome won't translate the E2 gene and then move to the E4. This is just a case where the ribosome could choose to translate this gene or this gene, and they are in different frames, which means that this entire region should be able to code 2 different frames. So we should see a mixture of frames within our signal, and our algorithm doesn't catch this very nicely. It has quite a large error. It will only capture about half of the E4 locus, but we can learn about the special signature. We see again that this signature, this is again 0 and +1, it's very different from this transition signature. So we have these 4 examples, we can learn from them, but in no means can we use just 4 examples to deduce about all other examples. And we want to do something systematic. We want to analyze, and in the human genome, we have almost 40,000 protein coding genes. We can't deduce 40,000 from just 4. So what do? We simulate sequences. So we take a sequence and we start introducing random mutations, but not just any random mutation. We want to introduce random mutation that would follow the coding table that I showed you, that would eventually result in the same protein. So we give penalties to mutations that would change the amino acid sequence, and rewards to mutations that would keep the amino acid sequence. And we generated a large grid of hyperparameters from number of sequences that we generate by mutation and the mutation rate. Okay, mutation rate has a strong influence on everything that we do. And we generated sequences having +1 frameshifts, -1 frameshifts, the dual coding region, like the HPV2, and just sequences having no frameshift at all to get some sort of measure of false positive rate, just to have the negative control. And we simulate a lot of these sequences, and then we use the calling algorithm that is rule-based to find the location of frameshift. And we see that our calling algorithm on the simulation is very, very accurate, even in this +1,0 dual coding region. So simulation is better than real life, obviously. But the main thing that simulation gave us is first more information about how frameshift would look like, about how dual coding regions would look like, and it would also give us a measure of success rates and false positive rates. So I told you that we simulated evolution to have a varying number of sequences from 5 to 19, including the original sequence, so it's 6 to 20, and a range of evolution rate. So this is how many mutation I would introduce. And these 3 tables would actually give me success rates, because this is how many times I was able to detect the frameshift that was simulated in the right location. And this little guy would give me the false positive rate, because this is how many times I detect some sort of frameshift when none really happened. And you can see that, well, success rate here is pretty high, but and the squares here mark the actual number of sequences and evolutionary rate that the actual data set that we analyze eventually has. So we can see that success rate here is quite low. This is very low because this is a very hard case. But false positive rate is also very high. About 30% of genes that we would predict to have frameshifts probably don't really have frameshifts. And this is a problem. This is a problem we have to face. And this is, I think, mainly due to the fact that for now, the calling algorithm is simply rule-based on observation. We had simulation, we looked at the simulation, we divided some features, and then we decided on rules. So it's very human-oriented. And now the question is, how do we move from rule-based to a completely automated classification? And this is the part that I've never talked about before. So rule-based is just a set of rules that is defined by the person who's writing the algorithm. It's defined on a small feature space because you're only human and you can't observe everything that is going on. And we want to do something that is bigger and better. And we can use a very large feature space, but this will call for strong feature selection because we don't want to overfit. Now the question is, which part should be automated? Should the inference of translational frame be automated, or should the calling algorithm be automated? And the obvious answer is both, but it's not that easy. So what I did actually do, which isn't in the thesis, but would be in the manuscript that will be published in a few months, is automate the inference part. So the feature that I choose to automate the inference part is the Fourier amplitude spectrum. I take this window, it has a periodic signal, I can just calculate the Fourier transform, and I can take all the amplitudes in all frequencies. Same for phase. I would use the inferred magnitude and phase as I calculated it, that I showed you before. I can also use the actual three vectors, but it's kind of redundant to use both because they talk to each other. Each observation in the space is actually one sliding window, so it's a lot of data because I have a lot of simulated data. It's about, I'll show you in a second, it's about 100 gigabytes of simulated data, which is thousands of these sequences. And some features are redundant, as I said, especially if you know Fourier transform, you know that when you calculate the Fourier transform, you have a multiplication of every frequency. You don't need the entire thing, you can only use half. So we need feature selection. We don't want to overcomplicate our model and running time. So the entire simulated sequence space is about 100 gigabytes large, and we need to do some balance sampling. So if we use the normal sampling, the normal division, which is 70 and 30 for validation and testing, we would get 15 gigabytes of validation and testing data set, which is not that bad because if you already have a model, you can load this validation and test set file by file and you run the classification. It will take the time it will take, but it's not a big data problem. What happens with the training? It's 70 gigabytes big, and we're in academia. We don't have supercomputers. We can't load 70 gigabytes of data to memory to create a classifier. So we need to use batch training. And batch training for neural networks, for example, is the normal thing that you do, but not for classic classification. And we chose decision trees. So a decision tree uses the entire data to create the nodes based on features and then create a classifier. We can't do that at once. We need to do something incremental, and it's not trivial. There are a few algorithms that do this. One uses what is called tree transposition, which is changing leaves into decision nodes and vice versa, but this would require us to keep all the data in memory somehow, or representation of the subtree in data somehow. So it requires data, a lot of data in memory. And there's methods relying on statistical representation of nodes, which is kind of like saving the best data, the data that was most influential on this node, and only using that in memory. An incremental decision trees is used for online classification, where you classify online, and then you can use the sample that you classified as another training example, and just keep incremental training. Overall, these, of course, feature selection could be very impactful because this is a decision tree. It's based on features. And overall on a stable data set, the performance here is pretty good. It doesn't affect classification that bad. And I can tell you that on the stage that we're on right now, which is not perfected yet, we get almost 97% accuracy for the simulation, and less than 1% of false positive rate. So we improved our model significantly, which is huge. The next question is, how do we automate the calling? So this is not a classification problem. This is kind of like, if you think about it in computer vision terms, it's kind of like segmentation problem. Because you might think of the different frames translated as different segments of an image. So if we were to use a neural network overall, everything might have been simpler, but much more complicated because it's a neural network. But how do we automate the calling technique in a classical, from a classical point of view? And I don't have the answer yet. I think it should be something like anomaly detection, because if you think about the classical translation, we don't have a frameshift. So if we have a frameshift, it's an anomaly. But I mean, if you have any other ideas, you can come talk to me. I don't think it would help because I don't have a predecessor in the lab anymore. So I don't know who's going to do that, but it's just interesting to hear your thoughts. So I'm going to end with two examples of frameshift predictions from the actual human genome, which are very interesting. This is the first one. It's a case. If there's anyone here that works on RNA modification, you would know the significance of this protein. And this is a case where you have a -1 frame shift close to the end of the protein. And I didn't mention this before, but this plot showing the frame also shows all the stop signals in the different frames. So the canonical frame would only have one stop signal at the end of translation, but the other frames might have stop signals everywhere because they're not supposed to be translated. What happens here, if we have a frame shift here, and translation would move on in this new frame, translation would probably move on beyond this stop signal. So we would have an elongated protein with a different sequence, a different domain at the end of it. And what happens for this specific protein is if we take the original protein sequence, I'm going to use bioinformatics words now, so I'm sorry. But if we're going to take this sequence and blast it against all other sequences now, we would get a lot of hits. They would be almost 100% identical, but the annotation would be low quality protein, which means that it's probably not a real protein. If we instead take the frame-shifted version that our algorithm predicts but doesn't appear to be annotated anywhere, so it's not a known protein sequence, and we do the same analysis, we would get, again, almost 100% identity, but for the major transcript of this protein. So this might mean that this is a mistaken annotation on the human genome, and there's another functioning version of this specific protein. What are the effects on function of this protein? We don't know. There is no known functional domain where the frameshift happens, but what we do know is that the secondary structure of this protein in the region of frameshift would change from an alpha helix to a beta shift. This, combined with the fact that the extra flank that this new sequence has a strong intrinsically disordered protein, I'm sorry if I'm talking complete Chinese, this combination would mean that it might be a protein-protein interaction site. This could affect the function of this protein immensely. It could cause it to be non-functional, or it could cause it to be functional in a different way. So this is just one example, and the second example is, I think, even more interesting. So this is what is called a GPCR. It's a protein that has a strong action of signaling within the cell. And what we see here is very interesting. We see that we have translation, we move to another frame, and then we go back. And if you remember, this signal is like the mixture of two frame signals. So it's not a complete frame shift, it's a mixture. And GPCRs in general are transmembrane proteins. They have 7 transmembrane domains. And they have an area which is outside the cell. So this is inside the cell, this is the membrane, this is outside the cell. This region would bind ligands. It would bind small particles from outside the cell. And these ligands would create some sort of signaling within the cell that would cause proteins to be active, inactive. It would cause a whole shenanigan within the cell. And the frameshift that we depict is exactly in this region of binding. And what happens upon ligand binding is that these transmembrane domain would change the conformation, and this would affect signaling. And in fact, when we check which ligands would bind to the sequence before and after frameshifts, these are completely different ligands. So this could completely affect this GPCR's function. And GPCRs are usually connected to cancer. This GPCR in particular has no evolutionary evidence to have this frameshift. It doesn't have a very close companion in other species, but it was documented to have a SNP, a single nucleotide polymorphism, which is a deletion, which actually creates a -1 frameshift very close to the positions that we predict. So maybe we have here an empirical evidence that this might happen. We don't know the implication. There's no annotation of disease or whatever, but it's evidence that this might happen. And we have a lot more examples. I don't have time to go into them now. It's very interesting. We gain signals, we lose signals, we get truncated proteins, we get proteins that lose degradation signals so they can become functional instead of being degraded. And overall, we have about 220 new proteins that we predict might be functional in the cell and have implications. Some of them are related to the immune system. Thank you very much. Do we do questions now, or?
So do you think these rare frameshifts occur as a fault, or is it naturally occurring phenomenon that we have?
We predict that this is something that is programmed to happen. This needs to happen sometimes. We assume that it might happen due to changing environments or due to exposure to pathogens, viruses, and whatnot, because this is usually a mechanism gained by viruses, bacteria, antibiotics, and stuff like that. So we think this is an evolutionary mechanism that was introduced to the human genome. We have evidence that it's a mechanism that is quite new, so we think it's quite new in evolution.
Yeah.
So we think it's programmed. It's not by mistake.
Is there a proof that there are proteins that transcribe from this function?
We have kind of a proof. We used ribosome profiling, if you know what it is, to kind of validate some of the frameshifts, and we get validation. So this is experimental proof that the ribosome could be in a different frame translating. So it is active.
But you cannot know if it's post-translational modification.
No, if the ribosome is in a different frame, then this happens during translation. So I can elaborate on that quite a lot. Maybe we can talk later. I mean, it's just how many, it's the simulated sequences. I know where the frameshift is, so I can test how many I catch and how many I miss. So this is the measure of accuracy. Of course, I didn't think you would have a question.
Is there a known mechanism to like a ribosome
Yes.
to shift left or right?
Yes.
And what's
So the mechanism, this is a tough question because the mechanism is known for -1 frame shifting and only on viruses. So it's hard to imply this on mammals, but we have hints of mechanisms in mammals, which is it requires a specific mRNA folding and mRNA secondary chapter and some sequence motifs that would allow this ribosome to get confused.
It's almost known, let's say, this mechanism.
It's hypothesized. It's known in viruses. It doesn't exist in our data set, but we don't expect it to. But we find something representative within the data set that we have, within our predictions. So we devised a mechanism which
I'm not asking whether this is happening, but how?
It happens due to folding of the mRNA, which causes the ribosome to not be able to move.
It detaches and then reattaches?
It doesn't detach or reattach. It just because the mRNA is folded in a specific manner, it can't move between nucleotides. It can move 1, 2, or just back. It's physical. It's a physical mechanism. Yes. For the mRNA. No, this specifically for this example, it doesn't have to happen. So, first of all, protein secondary structure prediction algorithms are not that great. They're not very accurate, and the untranslated regions, we don't expect them to have very structured. So it can't be a measure because if you take the translated regions and try to predict the secondary structure, you would mostly get junk. So it's not comparable.
What is the input and output of the classification?
Of the inference of the translational frame.
Yeah, what is the input to the model?
So
and
for the
output?
rule-based algorithm, it's just a variability vector. And for the decision tree, it's the Fourier spectrum, the Fourier magnitude
What do you
spectrum.
get out of the...
I get the frame of translation for that window. Canonical plus
So
1.
the input is the window
Yes.
and the output is the
you very much. Thank you.